Hi, I'm Paul Fuchs, and as Randy said, he and I and a group of others started the Center for Sensory Biology, which is sort of the nucleus of our presentations today. But out of that interaction has grown a number of, I think, very promising and exciting novel approaches in research. And so I want to just start by emphasizing the fact that at Johns Hopkins there's a very powerful ethos of collaboration. It's a place that even after 17 years there, I'm still amazed all the time by how generous my colleagues are and how easy it is to initiate new ventures and depend upon the resources of others to start those ventures. And so the Center for Sensory Biology is one expression of that, and from that has arisen other opportunities. So for example, what I have here on the board is a research group that Michael Caterina is the director of and put together to take advantage of some funding which is internal to Johns Hopkins, a call for novel strategies for research. And so in that endeavor, some funds were made available to the group of scientists that are listed here for a number of different research topics. As it happens today, we have with us Michael Caterina and myself, Dr. Raja and Matthias Ringkamp, all of whom are members of this pain work group. They'll be talking about aspects of pain, not necessarily the topics shown here, which are in their formative stages and just beginning to be done. But what I want to do is to spring from this kind of collaborative listing to tell you about how some novel approaches to the study of sensation and pain in particular have arisen from this sort of effort. So we're here today to talk about pain. My brother's dog knows full well what it means to tangle with a porcupine. She's done it many times. And yeah, it's amazing. She comes back with a face full of quills, and then 6 weeks later she does the same thing again. But this is very obvious kind of pain, and what I want to do is sort of take a slightly unusual approach and talk about a different kind of pain, which is personified here. Now, this is going to be a bit of a right turn from the main direction of our presentations today, so bear with me a moment. Let me set the stage. Every day when I come home from work, these two dogs greet me, and I bend down to pet them because they're so happy, and the little dachshund Heidi then barks in my face. And the response on my part is pain. It really hurts when she barks at me, that high-pitched, very loud, right next to my ear. I curse, I fall backwards, I hit my head, other kinds of pain ensue. But why does it hurt when she barks at me? And why is it so much worse now that I'm a bit older than it was when I was younger? So what I'd like to explore is the possibility that actually there's a very unique and distinctive neuroanatomical basis for what one might call pain in the ear. That is the enhanced sensitivity to loud sound, clinically referred to as hyperacusis. This is typically a problem which paradoxically also arises as people have partial hearing loss. So we'll perhaps be able to come back to that a little bit later. So what is pain in the ear? Well, let's begin first by just reminding ourselves about how hearing works. And there's just a brief video here that makes this point. I think. Oh, here we go. You won't be able to hear this, but this fellow on the left is just repeating a number of syllables whose sound spectrum and energy is profiled there in the middle of the slide. These sound waves propagate through the external and middle ears, setting into motion the middle ear ossicles, which are here, as you can see, responding to the vibration of the eardrum produced by those sound waves. That energy then propagates into the fluid-filled interior of the cochlea, the sensory organ that processes sound, where it sets up fluid waves within that organelle that then result in mechanical vibration of the sensory hair cells, which are located here on a flexible membrane. And these are the sensory hair cells here. And their little sensitive cilia on top are vibrated by the sound waves, which results in neural signals being transmitted to the central nervous system across a chemical synapse between the hair cell and the sensory neurons. And this process is frequency selective, enabling us to understand sound because the vibration pattern varies in position according to the frequency content of the sound. Then, because there are afferent neurons which selectively innervate hair cells along this mechanically tuned vibration pattern, they then serve as labeled lines, sending information about low frequencies, middle frequencies, and high frequencies to the central nervous system.